large about where we think, the greatest needs and opportunities and challenges are for our discipline moving forward in the next 10 years or next quarter century. So hope you left yesterday feeling excited, energized, and ready to dive in deeper today. We have two talks, one bookending the day, one at the beginning and one at the end, to kind of further spark your creativity, your thinking, and get your juices flowing for the kinds of stuff we're going to do for most of the day, which is taking the work we did yesterday to kind of define broad themes and specific questions within those themes to dig deeper into how we can craft them into kind of the full, basically, research agenda that we want to put forward to the community. So we're going to start with the talk and then have these breakout sessions to fill most of the day. And we'll give you more details on that as we move into that section. So thanks everyone across the planet where you are coming from later time zones and earlier ones for joining us today, and bring your all to it. And again, take advantage of the breakout sessions. For those of us who were in the lounge before this, it was quite a fun, engaging conversation and a chance to get to know some of the folks in this workshop. So take advantage of those breakout sessions for socializing and networking as much as you can. All right, we're going to jump in a little bit early just to give Kim her time, and then a chance for Q&A, and then as much time as we can for the breakout session. So I'm really excited to kick us off for the day with Dr. Kim Komatsu, who's from the Smithsonian Environmental Research Center. And she's going to be talking to us about leveraging power from multiple data sources to address long-standing and unanticipated ecological questions. So with great excitement, I turn it over to you, Kim. Please take it away.
Thanks. I think, Courtney, if you could stop sharing and I can start sharing. Great. Can you all see this? Excellent. Okay, great. So yeah, thank you so much, Ben, for the invitation to come and talk to everybody today. And I think, yesterday we had a really exciting discussion, and I'm looking forward to where we're going next. And I just want to say, for me, it's a real honor to have been invited to speak amongst this amazing panel of people who all have been so influential in really driving our field forward here in terms of synthesis. So thanks so much for the invitation. So I just want to start out by telling people who don't know who I am a little bit about myself. So I'm an experimental community ecologist. I've really been steeped in ecological networks since the very start of my career. So as a graduate student, I started up some NutNet sites, and I'll talk about the nutrient network a little more later in this talk. I've also spent the last 13 years working in the grassland system of the Kanza Prairie within the LTER network. And so really being part of these two different networks has me thinking very frequently about data synthesis. And I've also been really fortunate to have received funding from the LTER network and from NCEAS to host synthesis working groups out in Santa Barbara. So I've spent a lot of time out at NCEAS, writing on the walls in the offices and taking breaks to go up to the roof of the courthouse. And I'm really sad that we're not all in Santa Barbara today, especially because it's cold and snowy here in Washington, D.C. But hopefully altogether, this kind of gives you a little bit of context about the lens through which I'm coming at synthesis and the way I think about synthesis. And so just to start out, now that you know a little bit about who I am, I want to start by asking, again, what is the goal of synthesis? So when I'm thinking about synthesis, I'm thinking about looking across the planet and trying to identify generalities, but also find contingencies in the patterns that we're observing. And so I think, as was mentioned yesterday, we're basically asking, are there rules? And then what can we do with the rules that we might find? And really, I think it's important that we're not just considering the questions that we've identified in the past or studied in the past. I think we are moving forward in science, and so our understanding is hopefully progressing through time. And I think as a result of that, we should be changing our priorities constantly. And so there's really a need to utilize synthesis tools that are addressing unforeseen questions, things that might be critical for our understanding going forward. And so in the first half of the talk, I'm just going to kind of pontificate about the ways that I think we often go about conducting synthesis and what this means for looking to future questions. And then I'm going to delve into a case study example of how I think we can do synthesis going forward. And it's based in community ecology because at heart, I'm a community ecologist. So just starting out with this brief overview of how we've done synthesis in the past. So a very common method that was popularized starting in the early 90s is drawing from the published literature. And so ever since then, there's been a rise of these statistical meta-analyses, formal meta-analyses. And they focus on extracting the means and the variance around those means from tables and figures in the published literature. And then we statistically analyze them by looking at effect sizes. And there's lots and lots of resources that have been really helpful in teaching us how to do this. And they're a really great tool because they're allowing us to rigorously test hypotheses to identify generalities and potentially contingencies in the patterns that we're seeing. And there was discussion about this yesterday, so I'll go over it very quickly, but we also know that meta-analyses from the published literature have several limitations, right? So there's the publication bias that's widely acknowledged. Null results are often underreported in the literature. Differences in treatment implementation across studies can make it really difficult to compare results across studies. So Jessica gave the great example yesterday of fruit salad or applesauce, and what are we trying to look at here? And I think really an additional critical limitation here is that by drawing from the published literature, we're set up to be constrained by the past. So you can only pull information from published tables and figures that people thought to put into those tables and figures. And if we're developing new metrics, they might not be represented in these past papers. And so I'm going to leave behind pulling from the published literature for the rest of this talk and move into two other methods that we use to do synthesis now. So recently we've seen an emergence of coordinated distributed experiments. This is partly in reaction to the limitations of meta-analysis. And one of the first of these is the biodepth network. So this was set up with similar methodology to look at plant diversity effects on ecosystem function across sites in Europe. And of course, since then, there's been just a deluge of different types of new networks of coordinated distributed experiments. This is just a few of them shown here. And these coordinated distributed experiments are really designed to utilize consistent methodology across all of the sites. So they're trying to eliminate that problem of comparing different treatments across sites. But I think we know that the environment really differs around the world. So just an example of this map is showing estimates of nutrient limitation globally, and their darker colors are indicating higher nutrient limitation. So you think that's low soil nutrient availability. And so, for example, North American grasslands have much higher nitrogen pools than the Asian steppes. And so if we're designing an experiment and we're adding the exact same amount of nitrogen to sites in these two different locations, we might see differential sensitivity to those treatments, differential responses, because of that underlying nitrogen pool. And so that can really point to interesting patterns around the world. But when we think about utilizing this consistent methodology, we're actually applying treatments that might have different meanings across the sites. And so this is something that's important to consider. There's other cons potentially to these coordinated distributed experiments that are very similar to published meta-analyses. So there's bias in the volunteers who would join your networks for a variety of different reasons. Once you have volunteers, there's a relatively long time before you start to get the data to address the question you want to look at. And then I would say what I think of is kind of like the dirty secret of these coordinated distributed experiments is that people still don't follow these established protocols. So sometimes there's little deviations from the protocol, but sometimes there's really big deviations, and this can make it really difficult to compare across our sites still. But I think where these coordinated distributed experiments really shine is that they're moving away from those past constraints, because instead of just having the mean effect sizes, they have the actual raw data in big databases. And so the network might have been set up originally to address a specific question, but we can potentially reconfigure it to use it to address many additional questions in the future. And so then the third method of synthesis that I think is becoming more and more common is collating raw data from lots of different individual experiments into big databases. And so here, I mean, you come up with a question, you seek out all the people in the world who are doing an experiment that could address that question, and then you say, would you please provide us with your raw data so that we could do a synthesis with that information? And so we're kind of moving back to the idea here that these individual experiments are designed differently by investigators who have expert knowledge of their sites. And so that knowledge is really informing their treatment design. But that means that we have these differences in treatment implementation across studies that again can make them difficult to compare. We also, again, have a bias in the data availability. So in particular, older data sets might not be digitized yet. And you still need investigators who are willing to share their data with you. So there's an open access problem here. And as open access becomes more and more popular and kind of the culture of science now, it's becoming easier and easier to overcome this limitation, but it's still a significant problem that we should think about. But again, here, I think these databases of raw data from different experiments can really shine because we have that raw data in hand. And so again, we can reformat it or recalculate and address new questions with this data that we might not have thought of before. And so, of course, these approaches are not easy. Experiments are the backbone of all of these different databases, and they are very time and labor intensive. Bringing contributors to the data sets in is often very difficult to do, convincing people to join in. And then, of course, harmonizing data sets across investigators and across sites, no matter how good your input forms are, is going to be very difficult and very time consuming. And so I mentioned coordinated distributed experiments, assembled databases, both have their own benefits and limitations. And so I think once we've developed these networks, I would argue we shouldn't just limit ourselves to using just one or just the other. So I think what we should try and do is combine them together into these networks of networks. And hopefully we would do that in a way that takes advantage of the benefits of each of them and potentially leaves behind the limitations. We can use the wealth of existing data that we've spent so much effort to collect, and we could potentially ask new ecological questions that we might not have thought of before. And so hopefully the ultimate outcome would be that we're achieving this understanding that we couldn't find with just one data source alone. And so this sounds good. And of course, there's lots and lots of ecological questions that can be addressed with this type of approach. And so for the second half of this talk, I'm just going to walk through a case study example of utilizing this type of approach. And I'm going to use an example that I am personally interested in. So because I'm a grassland community ecologist, I'm going to bring us all out into the grasslands of the world. So this is just one example of one of my field sites. It's the mixed grass prairie of the central United States. I think it's beautiful. And as a plant community ecologist, I would go out in the field and I would put my quadrat down in this field of grass and then estimate the abundances of the species that I'm seeing. And so again, bringing you along for the ride with me, this is what you might see when you look at this field. At first glance, it might be difficult to pick out the species here, but if you spent as much time in the system as I have, you would immediately see species in this photo. So I'll point some out to you. So we have some rare species here. We have a scurf pea at the top, an aster species on the left, a white sage at the bottom, and a side oats grama kind of in the middle there. And in lots of ecological analyses, we really tend to focus on these rare species. So we're thinking about the total number of species in the system, the richness of that system. But these rare species really make up a very small fraction of the total plant cover in most of these herbaceous ecosystems. And rather, it's the abundant species or the dominant species in the system that I think are really critical to consider because they're contributing most to the functioning of that system just because of their abundance. And in this particular system, we don't just have one dominant species, we actually have two species. They're present in this mosaic. One I've highlighted in purple and the other I've highlighted in orange. And together, these two species are almost equal in their abundances, and they far, far outweigh all of the other species in the system. And so I would say this system is co-dominated by these two species. And codominance is a term that people have used really loosely in the literature so far. So most papers don't define at all what they mean when they say codominance, and those that do define what they mean have very, very different definitions. And so to try and address this issue, Jess Gray, who's a grad student at Colorado State University, as well as Mindy Smith and I have published a new Tansley review in New Phytologist. And we've come up with this synthetic definition of codominance. So we call codominant species that have shared abundances that can together comprise the majority of the total abundance of a community. And so you might be thinking, why should you care about codominance? And I would say there's 3 reasons, or if not more. First, codominated systems might be predicted to have greater stability of ecosystem function or ecosystem services because these species can compensate for each other. I would say when we're restoring ecosystems, we really need to know whether the system was dominated by a single species or multiple species. And then I think codominance can really help us understand basic ecological principles. So we can think about coexistence theory with the simplified example of codominated systems. And so that last one is the question that I want to focus on today. It's really a throwback to fundamental community ecology, thinking, does niche differentiation drive codominance? So just to delve into this a little more, because it's probably been a long time since Community Ecology 101 for most people. So we can think about basic coexistence theory here. And for plants, a primary niche axis along which species often separate is nitrogen availability. And so here what I'm showing is increasing nitrogen availability along this niche axis on the bottom, and 2 different species, an orange species and a blue species. And here I would say the blue species is the competitive dominant under low levels of nitrogen availability. So it has greater abundance than the orange species at those low levels, but the orange species can tolerate a wider range of nitrogen availability. And so we can add another axis onto here. Herbivore pressure is also thought to be a primary driver of plant community composition, especially in herbaceous systems. And so in this case, the orange species performs better under high herbivore pressure than the blue species. And so we can start to create these 2-dimensional maps of these species niches to find the environmental spaces in which they can exist. So we can make one of these for the blue species, one for the orange species. And when we combine these 2 niche axes, we would make the prediction that the trade-off across these niche axes is promoting codominance. So the space where these species are overlapping is highlighted by the black box. That's where they can coexist. And so because of this theory, we would expect that if we shift along either of these axes, we should be promoting competitive exclusion, or we would see the loss of codominance. And so we can test this hypothesis by looking at experiments that are either adding nitrogen or removing herbivores. And what I think is really important here is that we would predict that if niche differentiation is what's driving codominance at any given site, then the species that comes to dominate under these 2 different conditions should be 2 different species. So either the orange wins or the blue wins. And so how do we go about testing this hypothesis? Jess has developed what I think is a really clever metric that could be used to determine how many dominant species are in a system based on the definition that we came up with. And so what we do is we create a rank abundance curve. So we array our species in the community in order based on their relative abundances. And then we're calculating the difference in abundance between the consecutive pairs of species. And if the greatest difference in abundance occurs between species 1 and 2, then we would say the community has a single dominant species. If the greatest difference occurs between species 2 and 3, then we would say the system has 2 codominant species. If it's between 3 and 4, we would say there's 3 codominant species, and so forth. And so because this is a new metric, we can't go back to the published literature and see how this metric has responded to various different experimental treatments because no one's calculated it before. And so what we can do is really turn to these networks of databases of raw data of community composition data. And so for this example that I'm giving today, I'm going to utilize 3 different networks here. The first is the nutrient network. This is a coordinated distributed experiment. So again, investigators are following the exact same protocol around the world. And then I'm also utilizing 2 different databases that were assembled from raw data contributed from investigators running their own individual experiments, not coordinated together. So these are the core database. This was assembled by myself and Megan Avolio, and we're trying to look at community responses to resource additions. And then the Grazing Exposure Consortium, which was assembled by Sally Kerner. And the goal here was to look at the effects of large grazers. And so I don't really have time to get into all the details of these databases, but if you have questions about this, I would love to talk about it during the question and answer period. We've really learned a lot of lessons in assembling these databases, and they were really huge efforts to put together. So let me know if you have questions. But what I want to focus on today, importantly for this study, is that all 3 of these databases include experimental treatments that either added nitrogen or removed herbivores. And so many sites also applied both of these types of treatments separately as well. And so across these 3 databases, we're looking in herbaceous ecosystems around the world. They cover almost 500 different sites around the world. And so we can use these databases, again, of raw data so that we can calculate this new metric that no one has used before. And when we do that, what we find is that across all these sites, the vast majority of sites have communities that are dominated by at least 2, if not more, codominant species. Less than 20% of these sites have a single monodominant species. And again, here, I'm just talking about the number of codominants. And so this ignores all of the rare species in the community. But these communities, of course, have many, many more species than this. Okay, so knowing the distribution of codominance around the world, we can then ask what happens when we're either adding nitrogen or removing herbivores from these systems. So we calculated response ratios between treatment and control plots within each of our sites. And then we can compare the mean response ratios across all of our sites. And so starting out with our question of, are there nitrogen effects of nitrogen additions? So when we add nitrogen to such sites within the nutrient network, we see the predicted decrease in the number of codominant species within our communities. But in the core database, which is shown on the right here, we don't see this response. And we think this might be because the nutrient network has a goal as a network. Their goal was to completely remove nutrient limitation. And so they're adding really high levels of nitrogen in the plots to get rid of this nitrogen niche axis. And in contrast with the core database, we really don't know what the goal of individual investigators was. They're all doing their own thing and they're addressing their own questions. And they added varying amounts of nitrogen at all the different sites. But what we can do is separate out the core studies that added 10 or more grams of nitrogen to their plots versus those that added less. And then we do see a pattern emerge. So when nitrogen is added at comparable levels between the NutNet and core treatments, we see similar declines in the number of codominant species between these 2 networks. And so what we can do is combine these treatments together and draw from the power of both of these networks. And then when we do that, we see a decrease in the uncertainty or the variance around our estimate of this effect of the removal of that nitrogen axis on the number of codominant species. But then we could also say, what about these other studies in core that are not comparable in the amount of nitrogen added? Can we still learn anything from these? And of course, I think the answer is yes, we can always learn from data. And so here, I think what we could ask is whether there's a threshold to the effects that we're seeing. So within the core database, there's 7 sites that explicitly set out with the goal to address thresholds in nitrogen limitation. So they applied treatments at varying amounts of nitrogen within a site. And when we look across these 7 sites, we see that there is a threshold effect when you add nitrogen. So at about 8 grams per meter square, we see this line cross the zero intercept. And so it seems like adding 10 grams per meter squared in both the NutNet and core treatments is likely overwhelming these herbaceous ecosystems to remove that nitrogen axis. And then we see our decline in the number of codominant species. And we can broaden out to the entire core database. So here, the different sites added different amounts of nitrogen based on the investigator goals, but they're not necessarily different amounts within sites. We still see roughly the same pattern. So here, I really just want to point out that we can carefully consider the differences between our different networks. And because of that, we can draw from the power of an increased sample size when that's applicable, and really try and estimate what the true effect of nitrogen additions are on codominance around the world. But we can also learn from the differences between these networks. So again, we shouldn't leave any data out of our analyses. So then quickly, our second niche axis of herbivore pressure, I used the same workflow to combine comparable treatments across our different networks. This time I subsetted the NutNet treatments down to remove the sites that only had small herbivores. So we're comparing only the effects of removing large grazers between the NutNet, core, and GEX databases. And when we do this, we see the similar decline in codominance with herbivore removals shown on the right, as we saw with the nitrogen addition shown on the left. And so just to remind you of our hypotheses, we thought that we would, if we reduced either the nitrogen axis or the herbivore axis, we should reduce the incidence of codominance, and this is what we see. So when we remove these niche axes, we see a shift towards monodominated systems. And then our second prediction is that different species should come to dominate under these different treatments. And I don't really have a nice, elegant way to show this in one pretty figure on one slide, but I'll just tell you the answer. And the answer is this sometimes happens. So 78 of our sites included both nitrogen addition and herbivore removal treatments separately within these databases. And in 62% of those cases, we find different species come to dominate the communities under these two different treatments. And I think this is really interesting because it points directly to niche differentiation as the mechanism driving codominance in those sites. But then what about the other 40% of the sites? What's the mechanism there? And so potentially there's other niche axes we should be considering. Maybe these sites are driven by more stochastic processes. So I think there's lots of exciting avenues that we can explore with these databases that have many other experimental treatments in them that I haven't shown here today. So just to wrap up, hopefully I have convinced you that codominance is interesting to think about, and that codominance at many sites around the world is maintained by niche differentiation. And this is important because humans are altering our environment. We're often reducing niche availability. So we're doing things like removing large herbivores or polluting with nutrients around the world. And so we're altering these competitive structures of these communities. And by reducing codominance at these sites, we may be reducing the temporal stability of these ecosystem functions if these species compensate for each other. And then more broadly, thinking about synthesis in general, hopefully I've convinced you that we should not leave any data off the table. So combining networks of networks really provides us with the power to address these unanticipated questions that we might have. And we can also leverage network differences instead of thinking of them as limitations to provide us with additional insight into the questions that we want to ask. And so going forward with the future of synthesis, I hope that we can really draw on all of the available data that we have to try and find answers to the questions that we think are pressing or interesting in terms of ecology. And so with that, I would just say, thank you so much for listening. I'm excited to continue talking about synthesis for the rest of the day. And I really want to say thank you to the core GEX and NutNet network collaborators, people who are out there in the field conducting these experiments, providing their raw data, as well as all the funding sources that support these networks. And then I also want to say a thank you to the people who have helped me shape my ideas about synthesis throughout my career and also directly contributed to the ideas in these analyses today. And so I'll stop there, and if there's time, I'm happy to take questions. Otherwise, I'll be around the rest of the day if anyone wants to talk more about this.
Thank you so much, Kim. Unfortunately, we don't have a round of applause yet when you're in person, but yeah, we have a few questions already showing up in the Q&A. Folks, remember that's the great place to post them. We definitely have time for questions and answers, so I'm going to jump into that. I'm going to take moderator prerogative to ask the first one, and then I'll jump into ones that other people have posted. And so far, there's questions that kind of speak to the first part of your talk, and then some questions to the second part. And my question's a bit to the first part. So you framed out a couple different types of synthesis that you see. I'm wondering your thoughts on where or how the idea of idea synthesis that comes from different disciplines coming together to bring their ideas into the fold, fit into the sort of the structure you were laying out, and also other kinds of knowledge that are brought to bear besides through monitoring data or experimental data, and how you think about the landscape of synthesis with these other modes of bringing together knowledge, ideas, information.
Yeah, I think to answer both of your questions in one, the difficulty lies in talking about the same thing, right? So interdisciplinary work, we often use different terms to mean the same thing or the same term to mean different things. And so harmonizing our ideas about what we're even talking about can be very difficult. And then putting that data together that's often collected at different scales or for different purposes can be very difficult. And this goes the same way with using like these big observational networks. We're just often collecting data in different ways, and it can be hard to put them together. But that being said, I think it is very possible. And I again think that drawing from the differences can really help inform us in terms of why are we seeing differences between these different types of networks or these different disciplines? How can we put together all of this information in a way that informs us instead of just thinking, oh, this is a big limitation. We really can't address this. Let's not even try to put them together. So I would say that we should try to spend our effort talking to each other, learning from each other, and really making an effort to put these types of databases together. And that would lead to fruitful collaborations in the future, as opposed to kind of staying in our own little silos and working on our own things separately. I think we'll learn a lot more together. So hopefully that answers the question.
Yeah, great, thanks. So in that same space, someone asked, how do you ensure that investigator knowledge is considered when data are made open access? What if metadata are provided, but synthesis scientists don't go through them thoroughly or miss some important aspect?
Yeah, this is a great question, and I think it's really hard. So I'm glad you asked this because I kind of wanted to talk a little bit about the way we assembled the core NCEAS databases. And this is that we looked through the literature to find studies that addressed the types of questions we were interested in. We put out emails or tweets to our networks to say, hey, please provide us with information if you have experiments that do this. And then we got the raw data from the investigators. So in the vast majority of cases, they were emailing us data files. Sometimes they would point us to published databases that were online already. We also did go through those types of databases like EDI or Data1 to find additional data, but we always contacted the investigators. And so this, again, it takes an enormous effort. This is not an easy thing to do. But because of that, we were able to say, like, this notation doesn't make sense to us. How would this compare to that? And people were really invested in helping us understand their databases so that we could synthesize them in a way that was meaningful. Another point that I would make here is that part of the reason why people were so invested is because we offer co-authorship to everybody who provided data on all of these big synthesis papers that are coming out of these networks. So because these people are able to put their names on these papers, there's the incentive or the impetus that you really need to contribute to the ideas that are being formed here. So together, those two approaches, I think, providing authorship and also just being in contact with those investigators in the first place is a really good way to make sure that you're using the data in a way that is valid. In terms of just going to the databases that are out there, pulling out the data and then using it without talking to the investigators, I think it can be a really big problem that mistakes could be made in the metadata, either in the data that was provided or in the way that people interpret that from the investigator side. And so I guess I would just say being really careful in terms of scrutinizing all of those details and really making sure you are comparing things that are comparable is really important.
Yeah, great. Thanks. So a couple questions now more on the second part of your talk. I'm sure we'll return to the first part again with some other questions, but we have one saying, I hope this question is okay, but is there a specific reason that you don't have a single site in any of the huge grassland regions in Russia or former Russian regions? And this kind of speaks to one of the issues of synthesis is what if there's a bias in where things have been sampled and what does that do to the research questions you can ask?
Yeah, totally. It's an okay question to ask. And I think it's a really important question to ask all of us every time we do a synthesis, right? Like, where are we missing data? And in this case, the reason is we've asked people, and the people that we know, we've put out posts on Ecolog and Twitter again to say, if you have data, please provide it. But of course, we're missing people. None of the people who are leading these projects speak Russian. I know with NutNet, we've really had a big effort to try and increase participation around the world. But, you know, in terms of these coordinated distributed experiments, there are limitations to who's going to join the network. And some of those limitations have to do with the time and resources people can commit to doing something like this. It's not easy, it's not cheap. So none of these are all excuses. There's no good reason why we don't have data from that huge region. You probably noticed there's other big gaps in the map where there are grasslands. And all I can say is we've tried our best. We're all still looking for data. So if you know people, or if you can help us formulate in other languages requests for data, that would be wonderful. We'd love to add to these databases. But I think this is something we had great discussions about yesterday, at least in the group that I was in, and I'd love to keep talking about it today.
Great. A specific question about the results from your codominance work. So does the number of codominants correlate with system diversity?
Yep, great question. So of course, this is just like a tiny fraction of all the analyses we've done so far. We do see that at the site level, there is an effect of like gamma diversity at the site on the number of codominants. There's a positive relationship, as you would imagine. And then at the plot level, there's a lot of scatter. There's a very weak positive relationship. But here we're explaining like 2% of the variation by any broad predictor you could think of, like precipitation or gamma diversity or plot level diversity. We looked at productivity of systems. We really can't explain a ton of the global variation in codominance. So it's definitely an exciting thing to look into more in the future.
Cool, thanks. All right, two questions related to the databases. The first is, what are the main challenges or difficulties about how to combine different databases? And then the second one is, once these databases are assembled, are they open to the world or only to those that contribute? How do you balance the tension between effort and openness?
Great questions. So as with all synthesis, 90% of the time I've spent on this analysis is trying to harmonize the data sets together. And the challenges are different. So the databases themselves are very clean already to address questions that we've already looked at in the past, but putting them together, different treatments are called different things, all the different columns are named differently. There's information from some databases that's not in other databases, so we had to go back and find out things like what's the gamma diversity at a site we had to calculate for one of the databases. So even when within your database, your very clean data, you think you have everything, once you start putting them together, there's all kinds of things that you didn't think about. So I think, again, that's just a time and resource problem that we have to invest the time to be able to do this. The second question was really good, and I've already forgotten what it was. Can you remind me?
Yeah, no problem. The tension between effort and openness, like how do you deal with that?
Yeah, that's also such a good question. So in this case, again, individuals have to be willing to share their data. And some of these, within our assembled databases at least, some of the data sets that investigators are providing, they've said to us, we don't want this raw data published. You're welcome to use it. Every time you do a new analysis, please ask us about it. Other data sets within the database are already open. And so we've put a lot of effort into cleaning these databases, but we can't publish them because there's a subset of investigators that don't want the information published or made available. So what we're trying to do with this going forward, we do hope at some point in the future for both the core and GEX databases, and there's some discussion of this with NUTNet as well, to publish open access data papers that have all of this information, and we could just subset out the sites that don't want to opt into that. That's not a very satisfying answer in the spirit of open data, but I think that's kind of where we're at with the field, that not everyone is comfortable with this. But again, all three of these networks really try to give credit where credit is due. And so any paper published for any of these networks, people have the opportunity to opt in to contribute ideas and provide feedback on manuscripts or analyses. And we try to do this in a really rigorous way. So we don't just say, here's the final paper, we're going to publish it with your name on it, right? Like we involve people, here's the first analyses, what do you think? And then we revise the analyses, here's the next set of analyses, what do you think? And does this match what you see at your site? So really trying to engage people and give them credit through authorship is the way most of these networks are working, these three.
Yeah. Just going to do two more questions before we head to break. So one is actually, I think sort of in the same vein of trying to understand the nature of the databases you work with, but is asking, given that there are inherently some differences, sometimes big differences between the data that are being pulled together, what are some of the specific tools or approaches you've used to bring these data together into a global database? I think people are trying to get a sense of the challenges and the ways you've overcome those challenges in building these big consolidated data sets.
Yeah, so I guess I would like to say that we're like experts in database languages like SQL, and we could bring them together in a really nice way. And within NUTNet, actually, we do have great experts in that. And so their database is very well managed. But in terms of bringing them together, it's a lot of very detailed R code, trying to harmonize things, make it look right. Again, we've learned a lot of lessons. When Megan and I started the core database, we were like kind of by hand moving things around, and we've really moved toward R and this repeatable approach to combining data. And then I guess in terms of more statistical tools, I think thinking about the differences among these sites and what differences among the way people implement their treatments are between sites and how that might underlie the patterns you're seeing and give you spurious results is we use a lot of mixed models to try and incorporate these site level differences. So for example, I really glossed over this in this talk, but the size of the plots and the number of plots you have in your system really affects codominance. And that's just a methodological thing. And so we include those as random effects in these models to try and remove that variation and get at the underlying patterns. So really thinking through all of the methodological differences between these different databases and including that in your analysis to try and either highlight those differences and think why that's interesting, or remove the information that you're just not interested in for the sake of your question, I think is a good approach.
Great. I'm going to ask one last question. There's so many coming through, so we'll make sure you get them afterwards so you can respond to folks if you want. But there's a question about whether or not, well, so pointing out that there's quite a lot of nitrogen addition experiments in aquatic and marine systems. And just the question is, do you think that the codominance hypothesis can be generalized for these other systems as well?
Yeah, I think that's a really good question. I think I don't know enough about terrestrial or about aquatic systems to say definitively what I think the answer is, but I really am a proponent of the idea that patterns can be generalizable, and it would be really interesting to compare these across different types of systems. I mean, if someone has a database of community composition data in marine systems or freshwater systems, I would love to compare this across all of that. I think that would be so exciting. So I don't know, I guess is the answer. We should think about it.
Yeah, so research project. So.
And I guess I'll just put my email address in the chat. So if people want to email me questions, please do.
Perfect. All right, thank you so much. And yeah, sorry we couldn't get to everyone's questions. Wonderful talk. Thanks so much for the conversation afterwards, Kim. And we're taking a 15-minute break now. Please be back the half hour, wherever you are. I don't know what hour that is, but the half hour and 15 minutes. We're going to jump into the breakout sessions. So stretch your legs, get a coffee or a beer, wherever you are, and we'll see everyone back in 15 minutes. Thank you again, Kim.
Yeah, thank you so much.